In this video, we are going to be looking at eubacteria. Also known as true bacteria, these are prokaryotic, lacking a nucleus, cells. Also, they are the most common form of bacteria. We are going to be exploring the main phyla within this taxon and how to classify eubacteria accordingly. Also, we will be focusing on individual species within these phyla.
Eubacteria can be characterized by the following traits. They are prokaryotic, there is a lack of mitochondria or chloroplasts, they contain a rigid cell wall made of peptidoglycan. The flagellum, if present, is made of a single filament of the protein flagellin, unlike eukarya. They have a cell membrane composed of a phospholipid bilayer that lacks cholesterol and steroids. And finally, many eubacteria form spores resistant to dehydration and most temperatures, so when there is no food, they can last up to 50 years. Classification begins with three domains: archaea, bacteria, and eukaryota. These branch into their own 6 kingdoms, consisting of archaeobacteria, eubacteria, and plantae, fungi, animalia, and protista. Eubacteria, which we'll be looking at in more detail, then branches off into its 5 phyla, which consists of chlamydias, cyanobacteria, gram-positive bacteria, proteobacteria, and spirochetes. The first phylum we'll be looking at is proteobacteria. Proteobacteria are gram-negative and have an outer membrane made of lipopolysaccharides. Many move with the flagella and some are non-mobile. They include a wide variety of pathogens such as Salmonella, Vibrio, Helicobacter, and many others. Others are free-living, meaning non-parasitic, and include many of the bacteria responsible for nitrogen fixation. This is a process by which molecular nitrogen in the air is converted into ammonia or related nitrogenous compounds in soil. This is one of the largest and most versatile phyla. Proteobacteria can be found in virtually any environment across the globe, and there are some species in the phyla that can survive extreme environments with very little to no oxygen.
Some types of E. coli, or Escherichia coli, are harmless and even help keep your digestive tract healthy. But some strains can cause diarrhea if you eat contaminated food or drink fouled water. 75 to 95% of UTI infections are caused by E. coli.
Chlamydia are obligate intracellular bacteria. This means they lack several metabolic and biosynthetic pathways and depend on the host cell for intermediates, including ATP. Non-motile bacteria are bacteria species that lack the ability and structures that would allow them to propel themselves under their own power through the environment. Their outer membrane consists of LPS and membrane proteins. Chlamydia trachomatis STIs are commonly asymptomatic, meaning no symptoms. Evolutionary modification of the LPS structure likely evolved as a pathogenic strategy to silence innate host defense mechanisms.
Chlamydia trachomatis is an obligate aerobic intracellular parasite of eukaryotic cells. It is a gram-negative bacteria and it's coccoid or rod-shaped. It lacks a peptidoglycan cell wall, but rather contains the PG within the cell envelope, which plays a vital role in cell division.
The phylum Spirochetes contain distinctive diadem or double membrane bacteria, most of which are long, helically coiled, meaning corkscrew-shaped or spiraled cells. Spirochetes are chemoheterotrophic in nature, meaning they get their energy from consuming other organisms. The unique location of flagella is one which runs lengthways between the bacterial inner membrane and outer membrane in periplasmic space. These cause a twisting motion, which allows the spirochete to move around. And finally, most spirochetes are free-living and anaerobic.
An example of a spirochete is Borrelia burgdorferi, aka Lyme disease. Untreated, Lyme disease can spread to other parts of your body for several months to years after infection, causing arthritis and nervous system problems. It, however, doesn't cause problems in most small animals.
The gram-positive phylum is classified by the colour the bacteria turn in the staining method. The staining method uses crystal violet dye, which is retained by the thick peptidoglycan cell wall found in gram-positive organisms. This gives gram-positive organisms a blue colour when viewed under a microscope. Although gram-negative organisms classically have an outer membrane, they have a thinner peptidoglycan layer, which does not hold the blue dye used in the initial dyeing process. Gram-positive bacteria comprise of cocci, bacilli, or branching filaments.
An example of a gram-positive bacteria is Staphylococcus aureus, aka staph infection. Some types of staph normally live on people's skin and in the environment, and so don't cause disease. Other types of staph, however, particularly Staphylococcus aureus, can cause serious infections. You can get a staph infection from another person or by touching objects that have the bacteria on them. Staph bacteria that get in your bloodstream can cause problems in your organs. Doctors treat staph infections with antibiotics, but many staph infections are resistant to common antibiotics.
Cyanobacteria, or blue-green algae, are one of the most successful autotrophic organisms on Earth, which have mastered all types of environments, from freshwater, seawater, salt marshes, moist rocks, tree trunks, moist soils, hot springs, and even frozen waters. They are the most self-contained photosynthetic organisms, so are resistant and abundant. Their abundance can be gauged from the fact the Red Sea is named after the coloration provided by red-coloured planktonic cyanobacteria, known as Trichodesmium erythraeum. They are also one of the earliest colonizers of barren areas, and many cyanobacteria have the ability of nitrogen fixation.
Nosticals are an order branched off the phylum Cyanobacteria. They include filamentous forms, simple and branched. Nosticals include Anabina, which is a genus of filamentous cyanobacteria that exists as plankton. They are known for nitrogen-fixing abilities, and they form symbiotic relationships with certain plants, such as the mosquito fern.
We will now be looking at how Eubacteria can be classified into these different phyla. The only method of physical classification for this taxon is their shape. They can be spherical, or cocci, they can be rod-shaped, or bacilli, or spiral.
Often bacteria live on their own. However, in other cases, bacteria have formed clusters, staphylo, or long chains, strepto.
The first non-physical classification we will be exploring is classification according to how they get their nutrients. Eubacteria can be autotrophic, meaning they make their own food, which can be further split into photoautotrophs, which means they use light to make their food, or chemoautotrophs, meaning they use chemicals to make their food. On the other hand, they can be heterotrophic, meaning organisms that acquire food from other sources.
The next non-physical classification relates to how they use oxygen. They can be obligate aerobes, requires oxygen for survival; obligate anaerobes, does not need oxygen and dies in its presence; or facultative anaerobes, does not need oxygen but can survive in its presence.
The final method of non-physical classification of Eubacteria is known as gram staining. As we've previously discussed, this is classifying an organism based on the colour they turn with different stains. Although this appears an odd method for classification, it is actually one of the most important, as it can be used to understand how different infections can be treated. All eubacteria contain a peptidoglycan layer in their cell wall. That is made of sugar and protein.
Gram-positive bacteria are where the peptidoglycan is found in a thick layer on the outside of the cell. These bacteria show up purple or blue when stained. Gram-negative bacteria contain an outer layer that surrounds a much thinner layer of peptidoglycan. These bacteria show up pink or red when stained.
These two types of bacteria react differently to antibiotics, disinfectants, and release different toxins. This makes them fundamentally different. In a medical application, this is why you would treat gram-positive bacteria with penicillin, but not gram-negative bacteria.
That brings us to the end of our video on eubacteria. Thank you for watching.